Suppose here is sodium, sorry. Here is neurotransmitter, which is what? In our case, acetylcholine. Now listen with all your attention. When acetylcholine comes out, suppose here is a ligand gated ion channel, right? Whenever acetylcholine binds with this, ligand gated ion channel, this channel will now open, sodium will rush in. Now listen, as there are, maybe 20, 30 or 200 to 300 sodium channels or ionotropic receptors are open due to the, release of sodium, due to the release of acetylcholine in this small area. What will happen is this: suppose 200 to 300 ionotropic receptors are suppose open. Okay. Now a very important point here you need to understand is this: suppose 200 to 300 ionotropic receptors are open. Suppose a very important point is this: a very small amount of current will now flow through this area. A very small amount of current will now flow through this area. How can you measure it? It's simply use a probe, nothing else. Insert it here and keep the other side here. You will get. Now, whenever acetylcholine is released and it binds with ionotropic receptors, you will get a spike here. Okay, you will get a spike here. Now, this spike, this small spike, is actually not action potential, which is known by the name of EPSP. Okay, which is known by the name of Excitatory Postsynaptic Potential, or EPSP. Now, listen with all your attention. What is EPSP? EPSP is a small transient change in membrane potential disturbing the RMP, resting membrane potential. Now, a very important point here we need to understand is this: suppose this is your neuron. I am giving you three options. Okay, this is area A, this is area B, this is area C. From where action potential actually starts in a neuronal cell, where action potential start? Will it start from A, B, or C? The answer should be in between A and B, that is from here, this area. This area is way more important than you think. This area, that's why it has a special name, which is called axon hillock. Okay, it is called axon hillock. Now, why it is called axon hillock? You know that. Suppose this is your neuron. Suppose this is your axon hillock. Speciality of this axon hillock: it has got more amount of voltage gated sodium channel than this axon itself. Yes, that is the more important thing we should understand. Okay, that is, it has got more amount of voltage gated sodium channels in it than this patch of membrane. Clear? That means what I am saying is this: that whenever EPSPs, many EPSPs, now listen, suppose here one EPSP is produced, here another EPSP is produced. Now EPSPs are what? Small disturbances in RMP. Now here there is EPSP. Suppose here is EPSP. Okay. Now here there is EPSP. Now listen with all your attention. Due to these, EPSPs which are added, okay, this is summation, we'll talk about that later in this lecture. Whenever these EPSPs are added, now listen, this added potential difference will cause this axon hillock, sodium, voltage gated sodium channel to open, and more sodium will now rush in. Now, what is happening is, due to the EPSP, now EPSP is caused by sudden entrance of sodium due to the binding of neurotransmitter. In this case, here it is acetylcholine. Now, whenever sodium binds here, EPSP is produced, or excitatory postsynaptic potentials are produced. Now, this excitatory postsynaptic potential actually, start the conductance of sodium, and due to this conductance of sodium, there is a huge potential difference between this area of the membrane and this area of the membrane, and therefore these voltage gated sodium channels now detect the voltage changes, and they are now open in the axon hillock areas. And whenever they actually open these, small voltage gated sodium channels, whenever they will open these voltage gated sodium channels, a huge amount of sodium will now rush in, starting the process of action potential. Very important part is this: that means whenever these changes occur, then action potential will start from this area, and this action potential will travel. Why it is called action potential? Because it is a potential difference which can travel. A potential difference in action is called action potential. Clear? Now, this action potential will now travel here and cause the voltage gated sodium channel to open, so that this terminal branch can release the synaptic vesicles and neurotransmission can occur. Clear? What will happen, suppose, if instead of sodium or instead of acetylcholine we consider GABA or gamma aminobutyric acid, right? You know how to draw gamma aminobutyric acid? It's very simple. It's a butyric acid, right? That means it has got 4 carbons, right? Now this is alpha carbon next to the primary function group. This is beta carbon, this is gamma carbon, right? Now gamma amino, right? That's it. Add your H here, fill up the valences using H, We have already shown in organic chemistry class. Fill up the valences using H. Whenever there is nothing to do, fill up the valences using H. This is your gamma aminobutyric acid. This gamma aminobutyric acid is actually what? Inhibitory neurotransmitter. Inhibitory neurotransmitter. Clear? It's an inhibitory neurotransmitter. Now, because it's an inhibitory neurotransmitter, what is the difference between acetylcholine and GABA? Now listen. Because GABA is an inhibitory neurotransmitter, what will happen is, instead of sodium, chloride will enter. Chloride will enter. Chloride will enter. And what will happen is very important that you should understand that when chloride enters, then instead of EPSP, inhibitory postsynaptic potentials are generated. Inhibitory postsynaptic potentials are generated, and inhibitory postsynaptic potential generally do not cause this, voltage gated sodium channel to open. Why? Because inhibitory postsynaptic potential causes negative, charge potential, causes negative potential. Now listen, you now know, now listen with all your attention, you now know that why in the axon hillock action potential is produced always, all the time, why action potential is produced in the axon hillock. But a very important point you need to understand is this, there is another reason why axon hillock is the, controller of action potential. Now listen. Suppose this is your axon hillock area, okay? Suppose this is your axon hillock area, clear? Now, a very important point here we need to understand right now is this, that this axon hillock area has a threshold of minus 55 millivolt. A normal area of, membrane have a threshold of, normal area threshold is minus 35 millivolt. That means if you draw any graph, now suppose this is your minus 70, the resting membrane potential, this is your minus 55, suppose this is minus 35 millivolt. Now listen, suppose this is your, now this axon hillock have this, as threshold, minus 55 is the threshold. Now listen, suppose this is your, now suppose sodium ions now start entering the cell. What will happen is this, suppose there is no axon hillock, suppose this is a normal cell, then cell will have to reach minus 35 millivolt, otherwise action potential won't be fire. That is the rule. That means any cell will have to reach any threshold level. Suppose you are coming to my batch, okay, suppose two of you are coming right now, suppose, and I say that at least bring more 3 to 5 students, then I can start a batch, suppose. You know what is the meaning of this? That means my threshold is actually 5 or 7. That means if I get the remuneration of at least 7 students, I can actually start a batch. That means what? That means every cell, every neuron also has this type of remuneration thing. That means if I get minus 55 millivolt remuneration, I will start the action potential process. If you give me less than that, I won't be starting the action potential process and you will suffer. That means whenever sodium ions start entering, this try to reach this minus 55, and after that bam, it will fire action potential. Okay, we'll talk about that later. But if you give them sodium, if you give them less than minus 55, then you will get these spikes. Now what is this spike? What are these spikes called? These spikes are nothing but what? EPSPs. Now listen, therefore EPSPs alone cannot actually start the action potential because they are not strong enough. Okay, this is your threshold level. Okay, EPSPs alone do not reach the threshold level, therefore you are not getting 5 or 6 whatever it is, count here, you are not getting 6 action potential, but you are getting 6 EPSPs which are coming together, suppose, and they are actually initiating the process of action potential. Clear? Now what is the meaning of IPSP? During IPSP what happens is this, chloride enters. Now chloride can enter a cell when GABA binds with its receptor or glycine binds with its receptor. Now listen with all your attention. Inhibitory neurons in our central nervous system or in our body actually uses 2 types of neurotransmitter. That is, number 1 is glycine, number 2 is GABA. Clear? There is glycinergic neurons or GABAergic neurons. Both are actually inhibitory neurotransmitters. Why? Because whenever these inhibitory neurotransmitters bind to any cell or any ligand binding channels or ionotropic or metabotropic receptor, whatever it is, then chloride ions actually enter the cell, and therefore chloride ions make the cell more negative. This is your IPSP. Clear? This is your IPSP. That means whenever chloride ion enters the cell, IPSP is actually taking place. Therefore, no action potential will be fired because the cell is becoming more and more negative.